Welcome back to the deep dive. We're all about taking complex information, making it something you can really sink your teeth into. And today, we're continuing our exploration, picking up Chapter 4 right where we left off.
Exactly. We've built a solid foundation in the previous chapters, and now Chapter 4 takes us right into the critical neurochemical systems involved in psychosis.
Okay, so what's our focus for this session?
It's all about dopamine, glutamate, and serotonin neurotransmission in psychosis. A big topic.
Right, it sounds detailed. But our goal for you, the learner, is really to pull out the key insights. How do these brain chemicals actually work in this context?
Yeah, without getting totally lost in the weeds, we want those aha moments there.
Exactly. Find the connections.
Our mission here is basically to distill the most important stuff from this chapter. Give you a shortcut to being well-informed about the neurochemical side of psychotic disorders.
Good plan. And maybe before we dive deep, we should just quickly recap psychosis as the chapter defines it, just so we're all starting from the same place.
Good idea. So we're talking about symptoms like hallucinations, seeing or hearing things that aren't there, basically false perceptions.
The delusions.
Right, delusions. Those are firmly held false beliefs. Then there's disorganized thought and speech, and also abnormal motor behavior. That can range from agitation, sort of high energy.
Or the opposite, right, catatonia.
Exactly, catatonia, that state of immobility.
The text also mentioned a distinction with depressive psychosis.
Yes, it did. It notes that excitement might be seen in mania or schizophrenia, but depressive psychosis can present quite differently, more like psychomotor retardation, apathy, even anxious self-punishment or blame.
And it can be hard to tell that apart from negative symptoms sometimes.
Yeah, the chapter points out that difficulty in distinguishing them. It's tricky.
Okay, so with that framework, our mission for Chapter 4 is to understand the roles dopamine, glutamate, and serotonin play in how psychosis develops and how it looks.
That's the goal.
All right, let's unpack this, starting with dopamine. The chapter kicks off with the dopamine hypothesis of schizophrenia. What's the core idea there?
At its heart, the dopamine hypothesis suggests that hyperdopaminergia, basically an overactivity of dopamine in certain brain areas, is a major factor contributing to psychosis.
Okay, hyperdopaminergia, too much dopamine activity in specific spots. Now, before we hit those specific pathways, maybe just a quick refresher on how dopamine normally works, just basics.
Sure. So dopamine gets made inside neurons, then stored in these little sacs, vesicles.
Right.
When a nerve signal comes along, these vesicles release dopamine into the synapse, that gap between neurons. The dopamine then binds to receptors on the next neuron, passing the message along. And then, importantly, the signal gets stopped, usually by dopamine being taken back up into the first neuron. Reuptake.
Got it. Okay. That sets the stage. So the chapter highlights some key dopamine pathways relevant to psychosis. Let's start with the nigrostriatal pathway. Where does that run?
The nigrostriatal pathway starts in the substantia nigra. That's in the brainstem. And it projects, it sends signals to the striatum.
And its main job?
Primarily motor control, movement. And this is really relevant because blocking the D2 dopamine receptors here, like some older antipsychotics do, can cause movement problems that look like Parkinson's disease, drug-induced Parkinsonism. Yeah.
Okay, that's where the term EPS comes from, right? Extrapyramidal symptom?
Yeah, that's the older, less precise term, but yes. And conversely, if there's too much dopamine activity here, you can get hyperkinetic movements, too much involuntary movement.
Interesting balance needed there. Okay, what about the mesolimbic pathway? That one comes up a lot with psychosis.
It does. The mesolimbic pathway starts in the ventral tegmental area, the VTA, also in the brainstem, and projects to limbic areas. Think emotion, reward, especially the nucleus accumbens.
And traditionally linked to?
Traditionally linked very strongly to the positive symptoms of psychosis. The hallucinations, the delusions.
But the chapter suggests the view is changing a bit.
Yeah, it points towards newer neuroimaging studies suggesting the VTA and substantia nigra aren't totally separate. They're more like an integrated hub.
Okay.
So this hyperdopaminergia in schizophrenia might be better described as mesostriatal, affecting this broader network connecting the midbrain to the striatum, rather than just the limbic projections. It's a more interconnected picture.
Right, the brain isn't neatly compartmentalized. Makes sense. Okay, one more pathway. The mesocortical.
Right, the mesocortical pathway.
Yeah.
Also starts in the VTA, but this one projects up to the prefrontal cortex, our command center.
And it branches out there.
It does. One branch goes to the dorsolateral prefrontal cortex involved in cognition, executive functions, planning, working memory, that sort of thing.
Okay.
And the other goes to the ventromedial parts of the prefrontal cortex, which are more involved in regulating emotions and affect.
And what's the dopamine situation here in psychosis? Still too much.
Actually, here the hypothesis often flips. Many researchers think a deficit of dopamine activity hypodopaminergia in these mesocortical pathways might be behind some of the negative symptoms, like lack of motivation or flat affect, and also the cognitive symptoms, like problems with attention or memory.
Wow. Okay, so it's not just one simple story of too much dopamine. It's like too much in some areas, potentially too little in others.
Exactly. It seems to be about the balance across different circuits.
Fascinating. Now, this is where it gets even more interesting, I think. The chapter talks about dopamine autoreceptors. What are those?
Autoreceptors are basically self-regulating receptors right on the dopamine neuron itself. The chapter mentions D2 and D3 types specifically.
Okay. Where are they located?
You find them on the presynaptic terminal, the bit that releases the dopamine. Those are presynaptic autoreceptors. And also on the cell body and dendrites, those are called somatodendritic autoreceptors.
Their job
is feedback. When dopamine in the synapse binds to these autoreceptors, it signals the neuron to basically slow down. It inhibits further dopamine release.
Like a brake pedal.
Pretty much. The somatodendritic ones can also slow the neuron's firing rate. It's a way to keep dopamine levels from going haywire.
A neat regulatory loop. What about the dopamine transporter, DAT? How does that fit in?
DA, the dopamine transporter, is another key player. It's a protein on that presynaptic neuron, and its job is reuptakes sucking dopamine back out of the synapse and into the neuron.
So it stops the signal.
Right, it terminates the signal. And the chapter makes an interesting point about the prefrontal cortex here.
Oh, what's different there?
It seems there aren't as many DATs or D2D3 autoreceptors in the prefrontal cortex compared to other areas like the striatum.
Huh. And what does that mean?
Well, with less reuptake and less autoinhibition, dopamine might hang around longer and diffuse further away from the synapse. This could lead to something called volume neurotransmission.
Volume neurotransmission.
Yeah, like the signal spreads out more broadly, potentially reaching receptors that are further away. And in the prefrontal cortex, the main dopamine receptor is D1, which is actually less sensitive than D2 or D3.
So maybe this wider spread helps activate those less sensitive D1 receptors.
That's the idea. It might be a way the brain ensures D1 activation in the cortex, which is important for those cognitive functions we talked about.
That's incredibly intricate. Okay, let's switch gears now to another major player, glutamate. The chapter brings up the glutamate hypothesis of schizophrenia. What's the gist of that one?
So the glutamate hypothesis suggests that hypoglutamatergia, meaning reduced activity or hypofunction of glutamate signaling, especially at a specific receptor type called the NMDA receptor, is involved in psychosis.
Okay, so with dopamine, it was often too much activity, but with glutamate, it's potentially too little, particularly at these NMDA receptors.
Exactly, that's a key difference.
Now, the chapter goes into the glutamate-glutamine cycle. Sounds like a recycling program. Can you walk us through it?
Absolutely. It's a really elegant system involving both neurons and glial cells, specifically astrocytes, to keep glutamate available.
Astrocytes, the support cells. Okay.
Right. So first, glutamate is released from the presynaptic neuron, acts on the postsynaptic receptors. Then it gets quickly taken up, but mainly into nearby astrocytes.
How does it get into the astrocytes?
Through specific transporters called excitatory amino acid transporters, EAATs. They're part of the SLC1 gene family.
Okay, glutamate's in the astrocyte, then what?
Inside the astrocyte, an enzyme called glutamine synthetase converts the glutamate into glutamine.
Why convert it?
Well, the chapter suggests it might be a way to kind of protect that nitrogen atom, keeping it in the neurotransmitter pool rather than letting it get used for, say, protein synthesis. Keeps it dedicated.
Clever. So it's glutamine now.
Yes. Then this glutamine gets transported out of the astrocyte back into the extracellular space.
Using what?
Using other transporters, like specific neutral amino acid transporters, SNATs, and maybe another one called ASCT.
Okay, glutamine is outside the astrocyte. Where does it go?
It's then taken up by the presynaptic glutamate neuron.
Ah, back to the neuron that released the glutamate initially.
Exactly. And inside that neuron, another enzyme, glutaminase, converts the glutamine back into glutamate.
And then it's ready to go again.
Almost. It gets packaged into synaptic vesicles by the vesicular glutamate transporter, VGLUT, ready for the next release. It's a full circle.
Wow, that's quite the process. A very efficient recycling loop involving neurons and glia working together.
It really is. Keeps that crucial transmitter supplied.
Now, you mentioned NMDA receptors being central to the glutamate hypothesis. Tell us more about them. What makes them special?
NMDA receptors are a type of ionotropic glutamate receptor, so they form an ion channel. But they're unique. To open, they need two things to bind. glutamate obviously, but also a coagonist.
A coagonist, like a helper molecule.
Exactly. Either glycine or another amino acid called D-serine has to bind along with glutamate. Think of it like needing two keys to unlock the channel.
Okay, two keys, and when it opens...
When it opens, it allows positive ions, specifically sodium and calcium, to flow into the postsynaptic neuron, usually making it more excited.
You mentioned glycine as a coagonist. Where does the brain get glycine?
It can come from our diet, but it's also made right there in the brain by specific glycine neurons and also by glial cells, those astrocytes again.
Glial cells make glycine too.
Yep, and the chapter suggests that the glycine made by glia might be particularly important at these glutamate synapses.
How does glial glycine get to the synapse?
It seems to be released via a transporter called GLI-T1, working in reverse. Then, after it does its job at the NMDA receptor, the same GLI-T1 transporter takes it back up into the glial cell.
Another little shuttle system just for the coagonist. Amazing. Now, there was something else about NMDA receptors, a magnesium block?
Yes. The Mg++ block. This is really important. At the neuron's normal resting electrical state, there's a magnesium ion physically blocking the NMDA channel, like a plug in a drain.
Even if glutamate and glycine are bound?
Even if they're bound, the channel is blocked. But, and this is the cool part, if the postsynaptic neuron gets depolarized, meaning its electrical charge becomes less negative, that depolarization actually kicks the magnesium plug out.
So the channel only fully opens if the neuron is already a bit active and glutamate and the coagonist are present.
Precisely. It's voltage dependent and ligand gated. This property is absolutely critical for synaptic plasticity, the brain's ability to change and learn. It links activity level to receptor function.
That adds another layer of complexity and control. Okay, we've covered dopamine and glutamate. Let's bring in the third player, serotonin. The chapter mentions a serotonin hypothesis of psychosis.
Right. This hypothesis basically says that dysregulation of the serotonin system is also tangled up in psychosis and its symptoms. It's not just dopamine and glutamate.
Okay, another piece of the puzzle. Where do serotonin neurons live in the brain?
They mostly originate in the raphe nuclei, which are clusters of neurons down in the brainstem.
And where do they send their signals?
Pretty much everywhere. Serotonin neurons project incredibly widely throughout the brain cortex, limbic areas, brainstem, spinal cord. This broad reach means serotonin can influence a huge range of functions.
Makes sense. Now, the chapter highlights some specific serotonin receptors. Let's touch on a few key ones mentioned. 5-HT2A.
Yes, 5-HT2A. This one has a long history, especially because drugs like LSD, which cause hallucinations, act on this receptor. And importantly, blocking this receptor antagonism is a core mechanism for many antipsychotic drugs used today.
Okay, so strongly linked to perception, maybe hallucinations. What about 5-HT1A?
5-HT1A receptors. Partial agonists, drugs that partially activate them, seem to have anxiety-reducing or anxiolytic effects. They might also boost the effects of antidepressants, like SSRIs.
Interesting. And 5-HT1B and 1D?
For those, the chapter notes that blocking them or partially activating them might have procognitive potentially improving thinking and antidepressant effects.
Okay. How about 5-HT2C?
Blocking 5-HT2C receptors also seem to have antidepressant properties.
And the last couple mentioned were 5-HT6 and 5-HT7.
Yeah, the role of 5-HT6 is listed as unclear, still being figured out. But for 5-HT7, blocking those receptors might also be procognitive and antidepressant.
So quite a mix of potential effects depending on the receptor subtype. Now, the chapter introduces heteroreceptors, specifically talking about 5-HT1B receptors. What's a heteroreceptor?
A heteroreceptor is a receptor located on a presynaptic nerve terminal, but it binds to a neurotransmitter that's different from the one that neuron releases.
Okay, so it's listening for signals from other types of neurons.
Exactly. In this case, 5-HT1B receptors are found on the terminals of neurons that release norepinephrine, dopamine, histamine, and acetylcholine, particularly in the prefrontal cortex.
And what happens when serotonin binds to these 5-HT1B heteroreceptors?
It inhibits the release of those other neurotransmitters, norepinephrine, dopamine, histamine, acetylcholine. So serotonin acts like a brake on their release in the PFC.
Wow, so serotonin can directly dial down the activity of 4 other major neurotransmitter systems in the cortex.
Precisely. It's a key point of interaction between these systems.
What are the implications of that?
Well, if you block those 5-HT1B heteroreceptors, you might actually increase the release of those 4 neurotransmitters. That's why 5-HT1B antagonists are being explored as potential treatments for depression, for instance.
That makes a lot of sense. Definitely something to keep in mind for future discussions. Okay, so we've looked at dopamine, glutamate, and serotonin individually. How does the chapter describe them working together, interacting in psychosis.
It really emphasizes that they don't operate in isolation. They're deeply interconnected. Dopamine pathways influence serotonin release. Serotonin influences dopamine via those heteroreceptors. And glutamate signaling is crucial for the normal function of both systems. It's like a complex web.
And disruptions in this web could lead to the different symptoms we see: positive, negative, cognitive.
That's the idea. The mix of symptoms in schizophrenia likely reflects a complex pattern of dysfunction across these interacting systems. Maybe too much dopamine drive in mesolimbic circuits for positive symptoms, while too little dopamine in mesocortical circuits, plus maybe problems with NMDA glutamate signaling, contribute to the negative and cognitive issues.
But it's not fully mapped out yet.
Not by a long shot. The exact nature of these interactions and how they go wrong in psychosis is still a huge area of research. It's incredibly complex.
Understood. Now, near the end, there was a mention of schizophrenia potentially being progressive and something about NMDA receptors and synapse dynamics.
Yes, the chapter touches on this.
Yeah.
The idea is that ongoing issues with NMDA receptor function and glutamate signaling could affect synaptic plasticity, how connections between neurons strengthen or weaken over time.
Okay.
If critical synapses aren't properly strengthened during development, maybe especially during adolescence when there's a lot of brain reorganization.
Right. That's often when schizophrenia emerges.
Exactly. Those important synapses might get mistakenly pruned or eliminated. This loss of connectivity could disrupt information processing and contribute to the illness progressing. The timing of onset might be linked to this period of synaptic refinement gone wrong.
That's a really significant idea, linking it to brain development and maturation. Powerful stuff. Okay, before we wrap this chapter, let's quickly recap some of those key terms we defined.
Good call. We had hyperdopaminergy, too much dopamine activity.
And hypodopaminergy, too little.
And hypoglutaminergy, too little glutamate activity, specifically linked to NMDA receptors often.
Then the dopamine pathways.
The gross-radiatal motor, mesolimbic reward, positive symptoms, and mesocortical cognition, negative cognitive symptoms.
And the receptors on the releasing neuron itself.
Autoreceptor binds the neuron's own transmitter, usually inhibitory, and heteroreceptor binds a different transmitter, modulating release.
Got it. And the glutamate recycling system.
The glutamate-glutamine cycle. And the key glutamate receptor we focused on was the NMDA receptor.
Which needs a coagonist like glycine to open.
Perfect.
Great. So wrapping up our deep dive into Chapter 4, what are the really big takeaways for you, our listener?
I think the main thing is seeing how central dopamine, glutamate, and serotonin are to the neurobiology of psychosis, but also how interconnected they are. It's not just one chemical. It's the interplay between them across different pathways and receptors.
And understanding this neurochemistry is crucial, isn't it, for treatment?
Absolutely.
Yeah.
It's the foundation for understanding why current medications work or sometimes don't work well enough, and it's essential for developing better, more targeted treatments in the future.
It's definitely complex, and research is ongoing, but this gives us a really solid base.
For sure.
So maybe a final thought for you to chew on. We've talked about this intricate dance between dopamine, glutamate, and serotonin. Thinking ahead, what might the next big breakthrough look like? Will it still be about tweaking one neurotransmitter at a time? Or, as the field progresses, might we see approaches targeting multiple systems, maybe drugs hitting several targets, or even interventions aimed at tuning the circuits themselves, the way these systems interact?
That's the million-dollar question, isn't it? Moving beyond single targets to modulating the network.
Something to think about. Well, thanks for joining us for this deep dive into the neurochemistry of psychosis in Chapter 4. We hope you found it useful.
Hope to have you back for our next exploration.